The purpose of this video is to go over INTACT, which stands for Isolation of Nuclei Tagged in Specific Cell Types, and the way we perform this protocol in our lab. My name is Marco Bajic. I am a graduate student in Roger Deal's lab, and I will be going over exactly how we do this protocol. Briefly, INTACT is done on either fresh or frozen tissue, which is frozen, ground to a powder, spun down to isolate a pellet, which is then incubated with streptavidin-coated magnetic beads to isolate the nuclei using magnets. As long as you have a tissue-specific promoter, you can drive the expression of the nuclear targeting factor in different cell types. We can see the general first steps of INTACT shown right here, where we have all the different reagents we will need for this, which includes the mortar and pestle, the magnetic beads, nitrogen, the razor blade for cutting the protease, and the different magnets, as well as the ice and the tubes and the filters. So basically everything that involves the very first steps, which do go by rather quickly, is shown right here. So first we'll make sure that the spermine and spermidine are added to the incomplete NPB buffer, and that the protease gets added to that to make what we refer to as the nuclear purification buffer. So this right here is showing how the protease is being added into NPB. And once it's in the tube, we can actually grind this down further and add it to our NPB buffer, which is now complete. And then the same thing, except for instead of adding protease inhibitors, we are adding Triton for the NPBT. In this video, we're isolating nuclei from a whole root. And to do this, we're going to actually measure the amount of roots that we isolate, and when we collect these, we are putting them in the buffer. So right here, we're making one cut using a scalpel to separate the root from the shoot tissue. And then using forceps, we're just going to pull gently from one end of the plate to the other, grabbing the roots. And as you can see here, most of the roots are well collected. We get a weight to basically keep track of how many nuclei we isolate from specific amounts of tissue. Before actually going to the next step where we grind the tissue, you want to make sure that the final pestle will have the NPB buffer in it. And before starting to grind or freeze the tissue and grind it, we actually just will dab to make sure we get most of the liquid off. And using the mortar and pestle, the tissue is pulverized and ground to a fine powder, which is then scraped into the second pestle that contains the NPB buffer. So we don't actually add the buffer to the frozen pestle, we add the frozen tissue into the buffer in the other pestle. So to give a quick few grinds with the mortar, and then this is filtered through a 70 micrometer cell strainer. 70 micron cell strainer. Okay, and once all the liquid has made it through the filter into the tube, we are ready to spin. So we're going to do 8 minutes, 4 degrees in either 1200 Gs or 2000 Gs, depending on which tissue we're using. While the spin is happening, make sure to clean up your work area and prepare the streptavidin-coated magnetic beads. This involves taking the desired amount of beads, washing them with 1 ml of NPB, and to do this, we actually just use the magnet to pull the beads to the side after we've washed or stirred them a few times with NPB. And now we can just remove the buffer, and the beads, when we resuspend them, are in NPB, but they have been already washed once with a larger volume. So this just shows you the resuspension of the NPB. As the spin completes, you want to carefully and gently remove the tube, because you can see the pellet is right there at the bottom, and ever so slowly and carefully remove the buffer without disturbing the pellet at the bottom. Once this is done, you will resuspend the pellet with NPB buffer and add in the desired amount of streptavidin beads that have been washed. Okay, these are ready to go into the 4-degree cold room to rock for 30 minutes. Now, in that 30 minutes, you can prepare for the next step, which involve washing the beads that now will have the isolated nuclei. The wash steps involve adding 13 mL of NPBT. Into this, we add our 1 ml of the beads and the nuclei. Give this a few shakes. We'll actually put this on the rocker for 30 seconds. And then we take the tubes, uncapping the top to make the step when we come and collect the buffer easier without disturbing him. We incubate this on the magnet for 2 minutes. After the 2 minutes, we will remove the buffer, NPBT, without disturbing the beads, which are now on the sides where the magnets are. We go slowly and try not to disturb the beads. Okay, each wash step involves adding in up to 14 mils of NPBT, rocking again for 30 seconds, and then collecting on the magnet for 2 minutes. We do a total of 3 wash steps, and this demonstrates what that looks like. Again, the beads are on the sides, and when the final step is ready, we will use 1 mil of NPB to wash the sides of the tubes and collect the beads. But before we do the final collection, we actually take a 25 microliter aliquot out of the 1 mil resuspension, and to this we add in DAPI so we can visualize the nuclei and quantify them using a microscope. This just shows the addition of DAPI, which incubates at 5 minutes at the 4 degrees. Okay, and then with the remaining beads that have been washed, we will move that to a smaller tube. And now instead of them being in 1 mil, we want them in 20 microliters of NPB. So we'll use a magnet to collect the beads which contain our nuclei on the side of the tube, and then do a final elution and resuspension in the 20 microliters of NPB. And that's this just shows that process. So at the end, we have our beads, but to get the number of beads that are present within there, the DAPI stained nuclei will be visualized. For this, we use a hemocytometer so we can get accurate counts. Hemocytometer is loaded along the sides, and we use the DAPI fluorescence and bright field to visualize the nuclei and the magnetic beads, respectively. Here's an example of the beads. So right now we can visualize the DAPI fluorescence staining there as strong, and then the beads as the dark spots. And that concludes the general protocol for intact. Now you can freeze the nuclei at -20. However, I will point out that if you want to study chromatin architecture, it is best to use the nuclei immediately as opposed to freezing them. Some of the chromatin structure can actually change if you freeze the nuclei. If you're doing RNA work or just sequencing DNA, the nuclei can be frozen at -20 degrees for a long time. This concludes the video. If you have any questions, either leave a comment or email us, and we will try to get back to you. Thank you for watching.